All right, so we're talking this week a little bit about cell division. We're going to look at some processes and how our bodies generate new cells. All right, so one of the things we talked about previously was the cell theory, right? One of the postulates of the cell theory was that new cells arise from pre-existing cells, right? So there's this notion that, our bodies, our cells in our bodies do not have like an infinite lifespan, right? Our bodies are constantly regenerating our cell repertoire, making new cells, right? So the process of cell division is a very important process in living things where new cells are being generated, okay? So we have two processes, if you will, mitosis and meiosis, which allows our bodies to generate our cells, right? So mitosis is a process that's generating new body cells, right? We call them somatic cells. And then meiosis generates new sex cells, right? So for reproduction. So we're going to look today at two pathways or processes that our bodies use to generate new cells. All right, so mitosis and meiosis, they have the same purpose in the sense of generating new cell types, new cells, but we want you to be clear that meiosis produces one certain type of cell and meiosis produces a different type of cells, right? So remember, not all cells in our bodies are the same. All right, so we're going to look today at this process known as the cell cycle and how we generate new cells. All right, so again, just a little background information here. We've talked about it a little bit before, is that pre-existing cells, all right, cells that we already have in our bodies, divide to form new cells. Okay, this process is necessary in order for organisms to grow, in order for us to repair tissue damage, and in order for us to reproduce, right? We know that reproduction is a characteristic of life that we identified very early in the semester, right? Reproduction is necessary in order to continue to perpetuate a species, right? So cells contain genetic information in the form of DNA. We're clear about that. So when a cell divides, the information in our DNA, in the DNA of that cell, must be accurately replicated, and copies are going to be transmitted to the daughter cell through a series of steps. Okay. And so by the end of this lesson today, you should have a clear understanding of how the information that's contained in one cell, right, that DNA, how is that information duplicated, okay, and then prepared to be transmitted off to that daughter cell. All right, and so I'm using the term daughter cell, parent cell. So the parent cell is the cell that we start with, right? That's our sort of pre-existing cell. And when this cell divides and a new cell is created, we refer to that newly created cell as a daughter cell. Okay, jot that down in your notes. You've read, or you should have read through, I think we're in chapter 10, if I'm not mistaken. And so you probably see these terms, daughter cell, parent cell. So we're looking at how we take a pre-existing cell and create a new daughter cell. Okay, so a little bit more background is that the nucleus of eukaryotic cells contain large amounts of DNA. We're clear that in the eukaryotic cell, DNA is housed in the nucleus. All right, so over the next couple of days, we're going to look at how DNA, right, which is the genetic material, is packaged with proteins, and they form chromosomes, right? So you've heard this term chromosome before, all right? And we're going to look at two stepwise processes that allows us to generate new cells, right? So mitosis, jot this down. Mitosis is a process by which a series of steps allows for a parent cell to transmit genetic information to a daughter cell. Okay, so during mitosis, we are basically cloning. We're duplicating everything that's present in this parent cell so that it can be passed down to a daughter cell. That's what happens during mitosis. All right. However, meiosis is a little bit different. Okay. Meiosis is a reductive cell division in which the number of chromosomes are being reduced, okay, in preparation for sexual reproduction. So these daughter cells that are created in meiosis are going to be genetically different than that of the parent cell, okay, because the chromosome number is actually going to be reduced. All right. And we're going to take a look at that in a little bit more detail. All right. So what we're saying right now is that we're getting ready to see how the DNA, right, these chromosomes, how these chromosomes are duplicated and passed down to daughter cells, duplicated or reduced, depending on the process. All right. So what are chromosomes? Okay, jot this term down, chromosomes. These are the major carriers of genetic information in eukaryotes, right? These chromosomes are going to be found within the nucleus, right? And they're basically made up of chromatin fibers, which are basically a mixture of DNA and proteins, right? So these chromosomes are going to contain all of the genes that determine our features. And I think in another chapter or two, we'll go into a discussion of genetics and actually see the relationship between our genetic information, the genes in our bodies, and then the features that we actually exhibit, okay? So we do have a unit on genetics and heredity, maybe another chapter or two. So anyway, we have chromosomes in our bodies, right? So humans, write this down if you don't remember it. Humans have 23 pairs of chromosomes, okay? Normal humans have 23 pairs of chromosomes, okay? Each human cell, these are our somatic cells, they have 23 pairs of chromosomes, okay? You know, this 23 pairs of chromosomes is because we inherit a set of chromosomes from mom and a set of chromosomes from dad, okay? So 23 pairs of chromosomes mean that we have how many chromosomes in total in our somatic cells?
46.
46, thank you. And so this picture that I have down here on the right is a picture of a human karyotype, okay? Write down this term karyotype, K-A-R Y, wait a minute. Yes, O-T-Y-P-E. This picture of a karyotype shows us the alignment of the pairs of chromosomes in our bodies, okay? Our chromosomes are identified by a number, okay? Humans have 22 pairs, okay, 1 through 22, 22 pairs of somatic chromosomes and 1 pair of sex chromosomes, okay? Sex chromosomes are indicated by letters and not numbers, okay? And so we can look at this human karyotype, right, and identify a number of things about an individual, okay? The Human Genome Project has allowed us to map the human genome, right? And we know which genes are found on which chromosomes, all right? We know which genes are responsible for what, for various features of a human, right? We can look at these karyotypes and identify genetic abnormalities, right? So things like Down syndrome and other types of illnesses and syndromes that are genetic based. If we can look at the human karyotype, we might be able to identify some of those based on how things align with this karyotype. But what we're seeing here is that we see a normal human with 22 pairs of somatic chromosomes and 1 pair of sex chromosomes. All right. looking at this human karyotype and this pair of sex chromosomes here, that's XX, is this individual a female or a male? Does anyone know?
Female.
This is a female. Thank you. How do you know?
Guys have a Y chromosome,
Yes,
instead of two X's.
that is correct. So females have the designation XX for their sex chromosomes, and males have XY. And we'll come back to that in a minute, but absolutely. All right. So I mentioned the word genes. We've talked about our genes before. All right. So an organism may have thousands of genes, right? So we have over 20,000 genes that code for proteins. All right. A gene, by definition, is just a sequence of DNA that codes for a functional product, right? Those functional products are our proteins. All right. We've been talking endlessly this semester about the role of proteins in our bodies, right? We know that proteins are the functional units of our cells. Right? We heavily rely on this process of translation, transcription and translation for the production of proteins, right? And so our DNA serves as a template, if you will, for the production of these proteins, right? So we use genes. Genes are informational units. They're just sequences of DNA that each code for a functional protein product, okay? And so we refer to genes as informational units, as they provide information to carry out specific cell functions. All right. And so our genes, we know, govern specific factors, okay, such as our eye color, right? For the fly, the winglet of a fly is determined by its genes. The seed color of a plant, right? Our genes govern many of the physical features, right, about an individual, right? So again, when we get to our hereditary and genetics unit, we'll talk a little bit more in detail again about how our genes dictate our phenotype, okay, our physical features, all right? So we're talking here today, though, about these chromosomes, right? So we say that DNA is packaged into chromosomes. Okay, these chromosomes can vary in size and number depending on the species, right? So I showed you a picture of the human karyotype. We have a total of 46 chromosomes in our somatic cells, okay? The size of those chromosomes can differ, all right? So chromosomes number 1 through 22, they are all going to be of varying sizes, all right? For the human species, we're going to have a total of 46, but that number of chromosomes can differ depending on the species of the organism, okay? So a cat, a dog, a fish, these all may have different numbers of chromosomes, okay? So make sure you understand that concept, all right? And so structurally, this is what our chromosome looked like. Well, we have these two sister chromatids being held together by this centromere in the middle. You've probably seen this illustration of a chromosome before, all right? And so again, the point that I just made is that chromosomes differ amongst various species, right? We've talked about the diversity of life already. We know that there are millions of different types of organisms that exist. Every species have a specific number of chromosomes in their cells. Okay, so the number of chromosomes will not vary within a species under normal circumstances. Okay, humans have exactly 46 chromosomes in our somatic cells, normal humans. Now, let me ask you this. Are there situations where we don't find this number 46 in our somatic cells?
Yes.
Yes. Is that a good thing or a bad thing?
Bad.
Bad. Why is it bad?
Because then it causes genetic alters a genetic what it's called.
Yes, that's exactly what it causes genetic abnormalities.
Yeah.
Okay. It can cause genetic abnormalities, right? So for the most part, in this case, more is not better, and even less can be problematic, right? So we want to be in a situation where we have these 46 chromosomes, okay? genetic abnormalities can be the result of a deviation from this number 46, all right, depending on which chromosome is missing or there's an extra one. So Down syndrome, it comes to mind as I'm thinking about the trisomy. Number 21, we know that individuals with Down syndrome, you guys have probably heard about this before. These individuals have an extra number chromosome 21. Okay, too much. They call it a trisomy. There's 3 chromosome number 21 instead of 2. All right. And so we're saying here that humans have exactly 46 chromosomes in their somatic cells. All right. Other species can have different number of chromosomes. Okay. And so we also understand that information contained in chromosomes differ amongst species, right? So like I said before, we have the Human Genome Project that has mapped out the human genome. We know how many chromosomes we have. We know which information is contained on which chromosome. And that's what we know. Okay, but that may not be the same across all species, right? So let's just hypothetically say that the chromosome that determines our eye color, we'll just say is chromosome number 3. I don't know if that's true or not, but we'll say that the gene that encodes eye color is found on chromosome number 3 in humans. Okay, that may not necessarily be the same for a dog. Okay. Chromosome number 3 may not necessarily contain the same information on it as chromosome number 3 in the human. All right. So we're looking at a process known as the cell cycle today, the eukaryotic cell cycle. All right. 2 things I want you to be able to do about the cell cycle is being able to identify the stages of the cell cycle and be able to describe the principal events of the eukaryotic cell cycle. So we're looking at the eukaryotic cell cycle as the first step for generating new cells. All right. This is how we're going to create those somatic cells. All right. That process is known as mitosis. We'll come to it in just a second. So let's see the cell cycle. This is a process when this cell basically is ready to divide. Okay. When cells reach a certain size, we're clear that, again, these cells do not have an infinite lifespan, right? So when cells reach a certain size, they usually stop growing, right? Or they divide, right? Not all cells are going to divide. Some of them are simply going to die, right? But the stages for which a cell goes from one cell division to the next is called the cell cycle. The cell cycle is how we go from one cell to 2 new cells being generated. Okay. This cycle can take anywhere from 8 hours to 20 hours in plants and animals. Okay. It just depends on the organism. All right. This is a very important process that it needs to happen accurately with fidelity. All right. Because again, we're generating new cells. Right? We want these daughter cells to contain the correct amount of genetic material so that it can function properly. All right, so let's look at the eukaryotic cell cycle. You've probably seen this illustration before. Okay, generally speaking, the eukaryotic cell cycle consists of 2 main phases. Write these 2 phases down. We have interphase, right, which is collectively a period of preparation before it actually divides. And then we have M phase. Okay. M phase is where we have the process of mitosis taking place, which is the series of steps that allows this cell to divide into 2 identical daughter cells. Okay. So the 2 main phases of the cell cycle, interphase and M phase. All right. Interphase, we said that this is a period of preparation. All right. So if you look at this period right here from blue to red to purple, this whole inside that's gray is called interphase. Okay, there are 3 steps of interphase. Okay, you have G1, S, and G2. This entire phase is known collectively as interphase. Okay, there are 3 phases here. Okay, we have G1 phase, gap 1, S phase, which is synthesis phase, and then G2, gap 2. Okay, and then we have M phase. Okay, there are two things happening in M phase, right? This process of mitosis, right, where we're preparing for this nuclear division, right? And then the process of cytokinesis, right? Cytokinesis is when the cytoplasm of that parent cell actually splits off and becomes two new cells. Okay, so we're looking at the eukaryotic cell cycle as a process of preparation for cellular division. So interphase, right? As we're going to see, I have a couple more slides. We're going to go through this in a little bit more detail, but real quick, I want you to paint this picture in your mind is that certain things have to happen, right? Checkpoints, you know, growth, enzyme production, some things have to happen in interphase, right? Before this cell can prepare to divide in M phase, okay? So let's look at what's happening in each phase. Let's start with interphase, okay? So we're saying here that most of the cell's life is spent in interphase, okay? One important point to note here is that there's no cellular division taking place during interphase, okay? So if I asked you any types of question, the answer is always going to be false if there's any reference to cellular division happening during interphase, okay? This is just our period of preparation, all right? So no cell division is happening during interphase, all right? What's happening here is that the cell is growing. It is synthesizing materials that it needs in preparation for division, enzymes, growth, metabolism, all types of things that's making this cell nice and happy, preparing for division, okay? So G1, again, it's growing, it's metabolizing, it's synthesizing enzymes and things that it needs. And then there's this checkpoint here that's making sure that the cell is progressing the way that it should. And then it goes over into S phase, right, where DNA replication occurs. So remember, the goal here is to create two new cells. So in order to create two cells that are identical, we've got to duplicate whatever's here already. Okay, so during S phase, we're duplicating our genetic material. We're duplicating the number of chromosomes that are present. Okay, so that when we get around here to M phase, this cell can divide into two new cells, and both of those cells are going to have the same genetic material that parent cell started with, okay? So we're saying interphase is a period of preparation where the cell is growing, synthesizing the things that it needs. There's DNA replication. And so the steps of interphase, again, are going to include G1, S, and G2. I want you to know what's happening at each phase of, or each stage of interphase, okay? So G1 phase, what's happening at G1? This is an interval period during which no DNA synthesis is occurring, right? What's happening at G1 is just normal growth and metabolism, okay? These are processes that are gonna go on in the cell as usual, okay? That's taking place during G1, okay? This is typically, again, gonna be the longest phase of the cell cycle, all right? So normal growth and metabolism, the enzymes that are required, right, for this DNA synthesis to take place, these are going to become a little bit more active in preparation for S phase, okay, which is the next phase of the cell cycle, all right? So again, we've got normal growth and metabolism taking place at G1, all right? The next step is going to be S phase, which is the synthesis phase. Synthesis means to make, all right? We're going to synthesize or create more DNA, all right? S phase is synthesis. During this phase, DNA is replicated, right? Histones are made so that the cell can make or duplicate copies of the chromosomes that are present, right? So in a human, we've got 46 chromosomes in our somatic cells. So at the end of S phase, that number should be duplicated, right? So that when these two new daughter cells are created, they can each have 46 chromosomes, okay? So once this duplication is complete during S phase, the cell will enter another gap phase. Okay, she said, can I go back to G1? G1? Sorry. G1, okay. All right, so G1 is a period of growth and metabolism. All right, no DNA synthesis is taking place here. Okay, so we're producing enzymes and other things that are needed to enter S phase. Okay, but no DNA replication is occurring here. Okay, this is one of our interval periods. All right, so then we have S phase, which is a synthesis phase. During this phase, DNA is replicated. Okay, the number of chromosomes that are present will be duplicated. Okay, it is very important that this process happens with accuracy and fidelity. Again, if there are any abnormalities or deviations from the normal chromosomal content count at this phase, then we risk this cell either being signaled for death or these abnormalities will be passed down to the daughter cell. Okay. Next phase, after we've synthesized our new DNA, we've duplicated our genetic material now, we're ready for another checkpoint. All right, this gap 2 phase, right? This is now where the cell is sort of doing its checks and balances in preparation for division. Okay, this is a relatively short phase because all of the things that need to happen have hopefully happened at this point in terms of, enzyme synthesis, DNA is being synthesized, and now the cell is ready for cell division. Okay, so gap 2, G2 is gap 2, where the cell is checking to make sure everything has happened the way that it should, and it is ready for division. Okay, once G2 is complete, that is the end of interphase. Okay, after interphase is completed, the cell goes into M phase. Okay, M phase involves two main processes: mitosis and cytokinesis. Okay, mitosis, okay, is a stepwise process that, again, where these chromosomes are preparing for division. Cytokinesis is the actual process in which the cytoplasm divides into two new cells. Okay, this collectively is what happens during M phase. Okay, so as of right now, we have not divided this cell. All we've done was prepared the cell during interphase. Once we get into M phase, a couple of more stepwise processes need to happen, right? Which is basically going to separate these chromosomes into sister chromatids, and they're going to move towards opposite ends of the pole in order to prepare for that cytoplasm to divide into two new cells. So let's look at what happens during M phase. All right, so this is a schematic here that shows that M phase is a relatively short phase. Like, a lot has happened during interphase, right, to get us to M phase. All right, so we have G1, which was our growth period, right? Normal growth and metabolism. The cell spends majority of its time here in G1. All right, over here at S phase, we've got synthesis where our DNA is being reproduced. Okay, replicated, duplicated, whatever term you want to use here. And then G2 is another gap phase. It's making sure that everything has sort of happened the way that it should. And then the cell progresses into M phase, right? And so at M phase, the cell is almost ready for cytoplasmic division, right? But a couple more things need to happen, right? Mitosis is a stepwise process where these chromosomes are manipulated through a process of interphase, metaphase, anaphase, and telophase, which basically, again, is getting these chromosomes ready for nuclear division. All right, so let's look at the steps of mitosis. Okay, this is the first part of M phase. Okay, mitosis is a process that occurs over 5 stages. Mitosis is characterized by prophase, metaphase, anaphase, telophase, and cytokinesis. Okay, there's some things happening at each stage, right? During prophase, those chromosomes are going to compact and shorten and start to condense and become visible. All right, during M phase, you're going to see these chromosomes start to align towards the center of the cell. All right, during anaphase, those sister chromatids are going to separate now, right? And they're going to move towards opposite ends of the pole. All right, telophase is when we can visibly see 2 distinct sets of chromosomes at opposite ends of the cell. Okay, you're going to start to see these nuclear envelopes form around these 2 distinct groups of chromosomes, right? It is at this point now where we are ready for this cell to divide. Cytokinesis is where this cell actually splits off. The cytoplasm of that parent cell now splits off and creates 2 daughter cells. So we go from 1 cell to 2 cells during cytokinesis, the division of the cytoplasm. I'll give you a second to jot that information down. I know this is a lot, and I also have some images to illustrate what we just said. All right, so visibly, look at this, okay? During prophase, this is where we can actually see the chromosome starting to condense and become visible. We can see visible chromosomal alignment here during prophase, okay? And we have, again, we can see these sister chromatids here attached as chromosomes where we have these centromeres in the middle holding these sister chromatids together. During metaphase, you can visibly see these chromosomes aligning towards the middle of the cell. All right. I remember this metaphase with the letter M, metaphase for middle, right? We start to see these chromosomes lining up towards the middle or center of the cell, All right. During anaphase, we start to see the separation of these sister chromatids now. All right. They are moving to opposite ends of the pole, again, with the end goal of becoming this, because this cytoplasm is going to split. Okay. During cytokinesis. All right. So during anaphase, sister chromatids separate. They're moving towards opposite ends of the pole. And then here during telophase, we see two distinct sets of chromosomes, right, at opposite ends of these cells. And you start to see these nuclear envelopes forming around these sister chromatids, right? And during cytokinesis, you start to see this little indentation in the cytoplasm, okay, which is our indication that this cell is ready to divide. This is a cleavage furrow, right? This cleavage furrow forms as the cytoplasm starts to indent in the center so that it can eventually divide during this process of cytokinesis, right? Cellular division here will result in the creation of two new daughter cells that have the genetically identical content as the parent cell. Okay, so at the start of M phase, it is important that the process of growth, metabolism, replication of DNA, synthesis, that all of those things have happened with fidelity. All right, because again, now during M phase, these chromosomes are preparing for their equal distribution into opposite ends of this cell so that during cytokinesis, this cytoplasm can divide into two new cells. Okay. All right, this is another schematic that shows us mitosis or somatic cell division. All right, it's very important that you note that this process of mitosis, okay, is for the division of somatic cells only. Okay, sex cells are not included in this discussion right now. Okay, mitosis is for the division of our somatic cells. All right, we will come back and talk about sex cells on next class. All right, so mitotic division, we are going through the process of prophase, metaphase, anaphase, telophase, and cytokinesis, where we have the process of mitosis is complete. Okay, there are two cells with the same structures and chromosomes created, okay, as the parent cell. All right, this is just another schematic of the process of mitosis, right, where we have interphase, where we've got our periods of growth and preparation for division, and then we've got M phase, where we have prophase, metaphase, anaphase, telophase, and cytokinesis, where that cytoplasm divides. Okay, we're going to stop right here for today. What questions?